The overall goal of this experiment is to retrieve RNA-binding proteins of an RNA transcript from brown adipose tissue.
This method can help answer key questions in the adipocyte field, such as what are the regulatory mechanisms of non-coding RNA and the associated RNA-binding proteins. The main advantage of this technique is that it is easy to perform.
The RNA used to demonstrate this procedure is a fragment of the androgen receptor 3' untranslated region, hereafter referred to as the AR3' UTR. Begin by amplifying the T7AR oligo and a partial sequence of the firefly luciferase DNA from the SciCheck2 plasmid. The primers and parameters used are detailed in the accompanying document. Following amplification, use the PCR products as templates to synthesize biotinylated RNAs using an in vitro transcription kit following the manufacturer's instructions. Then, incubate the mixture at 37 degrees Celsius for 4 hours. After in vitro transcription, add 1 microliter of DNase to the reaction and incubate it at 37 degrees Celsius for 15 minutes to degrade the template DNA. Then, bring the total volume up to 60 microliters with water. Apply the biotinylated RNAs to purification columns to remove the salts and unincorporated nucleotides. Then, after measuring the RNA concentration, store the RNAs at -80 degrees Celsius for subsequent pull-down assays. On the day of the assay, thaw the frozen biotinylated RNA on ice. Then, to ensure that the RNA of interest has the proper secondary structure, heat 50 microliters.